Hey guys, in this video I'm going to go over everything you need to help you revise for your first OCR Gateway paper, Biology Paper 1. Now to go with this, to help you with this, there is a revision checklist, a revision guide you can download for free from my website, and you can tick stuff off as we go through it in the video. And then you can work out which areas you need to focus on, which areas you need to do a little bit of revision. Also on my website there are thousands of multiple choice questions to help you revise and predicted papers to help you study.
Microscopy techniques have varied wildly over the time. From the very basic starts where you had your lenses and you had to use the focus to see what was going on. These are all generally hand done, very basics. To ones that you're probably more familiar with in school, which have slightly more sophisticated lenses, to the massive ones that I used to work on, electron microscopes, where they're all controlled by computer. If you want to work out image height, object height, or magnification from an image you've taken from a microscope, the calculation is magnification equals image height over object height. We've heard of meters, which are incredibly long. You're probably between 1 and 2 meters tall. And we can have smaller parts of a meter. For example, a centimeter on my screen is about that big. And that is 1 times 10 to the minus 2 meters. A millimeter is even smaller. That's 1 times 10 to the minus 3 meters. A micrometer is 1 times 10 to the minus 6 meters. A nanometer is 1 times 10 to the minus 9 meters. And a picometer is 1 times 10 to the minus 12 meters. So if our meter is going to be m, our centimeter is cm, our millimeter is mm, our micrometer is mu m, nanometer nm, and picometer pm. As you can see, measuring very small things in meters wouldn't be a very useful way of measuring them. Even though you have to learn the structure of a typical plant cell or a typical animal cell, there isn't really a typical type of cell because there are a wide range of differentiated specialised cells. We can see here in our cross-section of the leaf, it has lots of different types of cells in. Here we have a neuron, which looks very different to a muscle cell, which is going to look very different to a skin cell or very different to a set of cells in the gut. They're going to be specialised to do their jobs. So here we have villi, which give us long surface area. Here the cells are very tall to provide structure. Here the cells have a very long body so that the neurons can travel a long distance. And the muscle cells are going to stretch and contract. Here we have our beautiful plant cell with a cell membrane. That's responsible for determining which bits go in and out of the cell. A cell wall, important for structure. The vacuole, important for structure. The cytoplasm, where most of the reactions take place. The tiny little dots are the ribosomes, which are responsible for protein synthesis. The green bits are the chloroplasts. The pink ones are the mitochondria, where energy is produced. And then last but not least, we have our nucleus. Here we have our animal cell with our cell membrane. Again, controlling what goes in and out, our mitochondria, where energy is produced, ribosomes, which are responsible for protein synthesis. Cytoplasm, where most of the reactions take place. And our nucleus, that's where the DNA is held, and that's the control centre of the cell. You'll notice there are several features of a plant cell that an animal cell doesn't share. For example, the cell wall, the vacuole, the chloroplasts. If you want to copy these pictures yourself, you can download them in the free revision guide from my website. Here we have our bacterial cell, which has its cell membrane controlling what goes in and out. The cytoplasm, where most of the reactions take place. The chromosome. The DNA, not in a nucleus. The flagella, which is used for locomotion. Ribosomes for protein synthesis, and then on the outside we have the cell wall. DNA is made from different bases that fit together. So we are always going to have A connecting to T, and we're always going to have C connecting to G. This is always going to be the case. It has a sugar phosphate backbone, and there are two of those which stretch all the way around the outside. There are two of those. It is a double helix. You see that the green is always connected to the yellow, A to T, C to G. The blue is always connected to the orange. And it's going round in a helical or a double helical structure. Amylase, protease and lipase are all enzymes and work with the lock and key mechanism. We have our enzyme, which has a very specifically shaped active site. So only one substrate or a couple of substrates are going to fit in there, the ones that have the complementary site. They're going to form an enzyme substrate complex, and then the enzyme is either going to break apart things or it is going to join together things. It is then going to release the products, and then the enzyme is unchanged and can be used again. You need to know how temperature affects enzyme activity, and it is this kind of lopsided curve. When we have really, really low temperatures, there is not enough energy. At the peak, this is the optimal temperature. And then after the peak, the enzymes get denatured, which means the links between them holding everything together are being destroyed. The enzyme is not killed. I know the temptation is to say this, but the correct term is denatured. Our curve for pH is much more symmetrical. We still have an optimal pH, but when it is too high or too low, the bonds aren't going to be in place. So the active site of the enzyme is going to be broken down. So again, it is going to be denatured. There are only a certain number of active sites on an enzyme. So once they are full up, the enzyme activity can't keep increasing. So while they are filling up, the enzyme activity will increase the substrate concentration, but when they are full up, increasing the substrate concentration won't increase the enzyme activity any further. An enzyme can be used as a catalyst for a rate of reaction. What we will see is that reaction will start to happen much faster, but it will end up at the same point. The reaction will probably end faster. This is because there are going to be other limiting factors like enzyme concentration, substrate concentration, or reactant concentration. For respiration, we are going to take glucose, add it to oxygen, and come out with water and carbon dioxide. You need to know the symbols for these. So oxygen is O2, water H2O, carbon dioxide CO2, and glucose C6H12O6. This needs to be balanced, but it's a nice easy one. 6, 6, 6. You have to make sure your numbers are the right size and in the right place. So these ones need to be little numbers and these ones need to be big numbers. Respiration is an exothermic reaction, which means energy is given out. The best example we can see of respiration is Screaming Jelly Baby demo, where we take potassium chlorate, that's our liquid oxygen, add in our glucose, that's our jelly baby, and you can see the massive amount of energy that comes off it. Anaerobic means without oxygen. So for anaerobic respiration, we take glucose and we turn it into energy and lactic acid. Not as much energy as aerobic respiration. Because the glucose isn't fully broken down. The lactic acid is going to build up in muscles, causing an oxygen debt. This build-up is going to be quite painful, so you'll get it when you're doing things like sprinting or when you've run out of oxygen. So after you've finished sprinting, after you've finished running to get rid of this oxygen debt, you're going to need to breathe really, really hard. That's why you pant. You keep breathing hard after you're running to pay back this oxygen debt, to get the blood flowing, to remove the lactic acid from your muscles. Anaerobic respiration can also take place in yeast. So yeast will take the glucose and will convert it into carbon dioxide and ethanol. Ethanol is used in drinks and cleaning products. And carbon dioxide is used for a variety of things, but when we're talking about in the context of yeast, that is what's going to make your cakes or your bread rise. There are a number of different enzymes in the digestive system that you need to be aware of. Lipase breaks down fats into fatty acids and glycerol. It is made in the pancreas and small intestine and works in the small intestine. Protease breaks down proteins into amino acids. It is made in the stomach, pancreas, and small intestine, and works in the stomach and small intestine. Amylase breaks down starch into sugars. It is made in the salivary glands, pancreas, and small intestine. And it works in the mouth and small intestine. You need to know how to test for fats, starch, sugars, and proteins. Fats can be tested for using the Emulsion Test or the filter paper test. For the Emulsion Test, you add ethanol, shake it, add water, and look for a colour change. If it goes cloudy, then lipids are present. With the filter paper test, if you rub it on filter paper, the filter paper should go see-through. To test for starch, you add iodine, and if starch is present, it is going to go dark black, dark blue colour. That means it's going to be a positive result. To test for additional sugars, we can add Benedict solution. Heat it for 2 minutes in a water bath, and if it goes red, if there's lots of sugar, or kind of like a pale green yellow if there's a little bit of sugar. We can test protein with the Buret's test, so we add Burette solution, and it will go purple if it is present. Photosynthesis is going to take water, carbon dioxide, and turn it into oxygen and glucose. We can take light and we can put it above the equation, but do not put it in the equation because it is not a reactant. It's just a condition that's needed. You also need to know the symbols for these. So water is H2O plus carbon dioxide, which is CO2, goes to oxygen, O2 plus glucose, which is C6H12O6. This needs to be balanced, but it's a nice easy one to balance because it is 6, 6, 6. So you can just remember that it's 6, 6, 6. And when you're writing out your formula, make sure your numbers are little and are in the correct place. Because if you write this, that's wrong, and you will lose the marks. In photosynthesis, we are taking energy from here, taking energy from light, and we are locking it up in glucose. This is an endothermic reaction. It takes in energy. There are certain requirements for photosynthesis. First of all, we are going to need chlorophyll. That is our green pigment in leaves. We're going to need water and carbon dioxide because they are our reactants, and then we're going to need sunlight. And the levels of these can greatly affect how much photosynthesis takes place. The rate of photosynthesis is going to depend on the percentage level of carbon dioxide. As the percentage level of carbon dioxide increases, so the rate of photosynthesis is going to increase, but only up to a point. After this point, there are going to be other limiting factors. Past this point, we need to increase something like the water, light, or the temperature if we want more photosynthesis to take place. We could easily switch this out to the percentage level of water and the graph would look the same. Light intensity is important for the rate of photosynthesis. When it is nighttime, when it is dark, we do not have lots of photosynthesis going on. As we get further through the day, as we get more light intensity, the rate of photosynthesis will increase until we get to a point where it is no longer the limiting factor and other things like the reactants or temperature need to be increased. After this point, we need to think about increasing other things. Now, even though the graph is flat here, it looks like it might have stopped. It hasn't. There is still a steady rate of photosynthesis. It's just not increasing as much as it was down here. It's just a steady rate. When plants are very, very cold, everything acts very, very slowly. Not a lot happens. It slowly increases until a nice point where the enzymes are happy and there's lots and lots of photosynthesis going on until it gets too hot and they start to be denatured, and then the rate will fall off very rapidly. So we have our rate of reaction increasing with temperature and our optimal temperature and our enzymes getting denatured. It's really important that you remember that the enzymes are denatured. They are not killed, they are denatured. The actual rate of photosynthesis that takes place is much more complicated than depending on just one thing. It's going to depend on lots of different things all at once. The glucose from photosynthesis is going to be stored as starch. The most obvious example of starch is going to be potatoes. You are doing such fantastic work. Well done for making it this far. We're just going to take another little mind pause, another little break for you to gather yourself, refresh yourself, and then we're going to start again. When we're talking about diffusion, we are talking about things moving from a high concentration down the diffusion gradient to an area of low concentration. This could be things moving from an area inside a cell where they've been made to another area, or it could be things moving out of a cell. For example, it could be happening in the lungs. These are the alveoli, the air spaces, and this is the capillary travelling around it. These are very, very thin, walls only 1 cell thick, and carbon dioxide is going to diffuse from the blood into the lungs so that it can be breathed out, and oxygen is going to diffuse from the lungs into the blood so it can be taken around the body. All this can be in the gut. These are the villi of the gut. This is the gut cavity here, and you notice again they are 1 cell thick, and just like the alveoli, they have a very large surface area. We're going to get digested food moving from the gut cavity into the blood so that it can be taken around the rest of the body. So diffusion is the movement of gases or any particles that dissolved in solution moving down a concentration gradient from a high concentration to an area of low concentration. Osmosis is specifically the movement of water through a partially permeable membrane from an area of high water concentration to an area of low water concentration. So you notice this partially permeable membrane, the pores in it aren't large enough for the solute to move through, so the water is going to be the one that moves through here. This sort of thing can happen in root hair cells where we're looking at the uptake of water. Active transport, again, is the movement across a membrane, but it's from, this time, a low concentration to a high concentration against the concentration gradient. So our channel, or active transport channel, is going to pick up something that it wants. It is then going to move that through the channel to the other side. This could happen, for example, when we're talking about glucose in the gut or minerals in roots. In mitosis, we go from 1 parent cell to 2 identical daughter cells. The first thing that needs to happen is that the DNA in the nucleus needs to condense into chromosomes, and then they need to line up down the middle. Once they're all lined up down the middle and all the checks are taken place to make sure that chromosomes aren't going to go astray, they can start to be pulled apart to either end of the cell. New nuclei will form, and then they will separate into 2 identical daughter cells. All cells start off looking the same, so they have your basic cell structure, and then various different genes will be turned on and turned off. And that's when it will start to specialise. That's when differentiation will take place, and it will grow this really, really long axon, or it will grow the villi, or it will turn into a leaf cell. Stem cells are fantastic things because they are things that have the potential to turn into any other type of cell. They have a number of different uses. For example, if you're treating Parkinson's disease, they can be used to grow new brain cells. If we're talking about brain or spinal injury, bone injuries, then they can be used to grow new bones to fill the gap. If we have organ failure, we can grow new organs or parts of organs instead of waiting and making someone wait on the incredibly long transplant waiting list. If we want to make stem cells, then we take a nuclei out of an egg cell. We take nuclei from the patient cell and insert that into the empty egg. The egg can then start to develop into an embryo. From this embryo, the stem cells are then removed and stem cells are turned into new cells. This does come with quite a lot of controversy because human embryos are going to be created and then destroyed, and there are lots of religious objections to this, people just saying that life starts when embryos are created, and people that object to the destruction of embryos. Here we have our respiratory system. Air goes in through the mouth or the nose, down into the trachea, which is also known as the windpipe. Then into the bronchus, which is a branch of the trachea, into the bronchioli, which is a branch of the bronchus, and into the little grape or cauliflower-shaped alveoli. This is where gas exchange happens, and they have an incredibly large surface area. Your diaphragm moves up and down to bring air in and out. The heart pumps blood around the body. The intercostal muscles allow the rib cage to expand. And the ribs, the last part that makes up everything, protects the lungs. Here we have a cardiovascular system, and it is a double system. The blood gets pumped from the heart to the lungs, goes back to the heart, and then gets pumped around the rest of the body. If you see a picture of the heart, the first thing you do is write right and left on there. We have our vena cava, where the blood enters. It goes into the right atrium, down through a valve into the right ventricle. From the right ventricle, it goes up and to the lungs via the pulmonary artery. It comes back into the heart via the pulmonary vein, into the left atrium, into the left ventricle, and then is pumped to the rest of the body via the aorta. If you want to check you have the path of blood right, then we need to be looking at capital letters. It goes through the vena cava, the atrium, the ventricle, then the artery, back through the vein, into the atrium, to the ventricle, and then the aorta. So it goes vena cava, atrium, ventricle, artery, vein, atrium, ventricle, aorta. VA, VA, VA. If you don't have that pattern, you've made a mistake somewhere. Other features of the heart that you need to know are here. These are valves. They will only allow blood to flow, and that this side has a much larger muscle than this side. The right side only needs to pump blood to the lungs, which aren't very far away, but this side has to pump blood to the rest of the body a much longer distance. The majority of the time, veins carry deoxygenated blood apart from the pulmonary vein, which carries oxygenated blood back into the heart. And the majority of the time, arteries carry oxygenated blood apart from the pulmonary vein, which carries deoxygenated blood from the heart to the lungs. If the heart isn't functioning properly, pacemakers, artificial pacemakers, can be introduced to help the heart keep time. Or if somebody has cardiovascular disease, then these tubes can get blocked up. Blood is made up of several components. The actual colour of blood is this pale yellow colour. This is the serum. That's the liquid component of the blood. The cells give it its actual colour. Red blood cells, the cells that give blood its colour, have no nuclei. And this is so they have more space to carry oxygen, which is their main function. White blood cells are part of the immune system. And platelets are fragments of cells, and they are important for things like clotting. Arteries have a very thick walls because they are carrying blood under high pressure, which means they have a thin lumen. That's the gap in the middle. Capillaries are very, very small. They are only 1 cell thick, or very, very thin, I should say. They're only 1 cell thick. This is to allow for diffusion. They generally go around in this kind of like mesh network around things like the gut, around the villi in the gut, around the alveoli in the lungs, so they have a large surface area. The veins carry deoxygenated blood. They carry it back to the heart, so they have valves. And they have thin walls and a thick lumen because they're carrying blood under low pressure. Here we have a cross-section of a typical leaf. Our palisade mesophyll, where photosynthesis is going to take place. Cuticle, which is the waxy layer. Upper and lower epidermis, which cover the plant. Spongy mesophyll, which is a space for gas exchange. And the guard cell and stomata, which is where transpiration takes place. Inside the plant we have the xylem and the phloem. The phloem is going to carry water. This is generally going to be an upwards direction from the roots where it is collected to the leaves where it can be used for photosynthesis, and the phloem, which carries ions and food, and this is generally in a downwards direction from the leaf where food can be made in photosynthesis to the roots where it can be stored in, for example, potatoes. There are several factors that affect the rate of transpiration, and transpiration not only involves water moving out of the stomata, but also moving up through the xylem. So if we have bright light, that is going to lead to more transpiration. More light means more photosynthesis, which means there's going to need to be more water brought up into the cell. If we have a high temperature, that is going to lead to more transpiration. Because the rate of reaction is going to happen faster. If we have high wind, that is going to lead to more transpiration. Because wind is going to be brushing against the leaf or flowing against the leaf, moving things out of the way, so diffusion is going to play a part here. And if we have high humidity, this is going to lead to lower transpiration. Water is going to struggle to leave the leaf because there is a very high concentration of water. It's very humid outside. Here we have the male and female endocrine system. The pituitary gland is in the brain. Thyroid is in the neck. The adrenal glands are on the kidneys. Pancreas is hiding behind the stomach. Ovaries are kind of like hip level. And testes hang below the penis. The testes produce testosterone, which has the effect of growing muscles, making the balls and penis drop and grow larger, increasing the rate of hair growth. Estrogen is produced in the ovaries. That's responsible for the maturation of eggs and the menstrual cycle. The pancreas produces insulin, which is important for regulating blood glucose levels. The adrenal glands produce adrenaline, which is important for our fight or flight response. The thyroid produces thyroxine, which is important in regulating our metabolism. The pituitary gland is very busy. Among other things, it produces follicle stimulating hormone, FSH, and luteinizing hormone, LH. One of the reasons periods feel so rubbish is because your hormones are literally all over the place. Starting with follicle stimulating hormone, it rises, peaks, and its job is to make the small egg grow up to a larger egg and then be released. Luteinizing hormone is only active for a very, very short period. Its job is to release the egg. Estrogen builds up until it stimulates luteinizing hormone. Progesterone builds up slowly as it builds up the lining of the uterus. And if there is no if there is no embryo implanted in it, that will decrease and the lining of the uterus will break down. There are a number of different methods of contraception, some hormonal, some non-hormonal, that will stop you getting pregnant. But not all of these will protect against sexually transmitted diseases, so it's always very, very important that you wear a condom. This is a barrier method of contraception. This will stop the sperm getting the woman pregnant, and it will also stop any nasties that are going between transmitted from her to him or from him to her. The pill and the coil, IUD, intrauterine device, are hormonal methods of stopping getting pregnant. They are going to stop the egg being released or the egg being implanted. The diaphragm is a barrier method because it will stop sperm entering the vagina, but the semen will still be transferred into the vaginal entrance so that you can still get sexually transmitted diseases this way. If you're sure that you don't want to have children, you can go to be sterilised, you can have a vasectomy, you can have your tubes tied, which will mean that no sperm will get from the testes out to the penis, or for the woman, no egg will be released. Around 1 in 6 people will find themselves in the unfortunate position where they can't have children naturally. About half this is due to male-related reasons and half this is due to female-related reasons. As you can see, I am one of those people, and last year, 2016, we did IVF, and this is my massive bump. So the obvious advantages for IVF are you get a baby out of it, and if you've been in a situation where you can't have something that you really, really want, you know it's very, very sad and affects your mental health quite a lot. So having a baby is going to be good for people that want to have a baby, their mental health. However, the disadvantages are you have to take a large number of drugs for a very, very long period of time. These have very nasty side effects. As well as the daily injections, which leave you horribly bruised. There are long-term consequences for these. Because taking these IVF drugs increases your chance of various different types of cancer. It is very, very expensive. I had to have it twice. That's twice as expensive. Doesn't always work. There is about a 40% success rate with IVF, with each round of IVF costing a minimum £5,000 with a 40% success rate. Here are all the large number of drugs that I had to take day by day. It's a very costly, time-consuming, painful process. Homeostasis is the maintenance of a constant internal environment. And to keep your body functioning properly, we need to control our blood glucose levels, our water levels, and our temperature. The brain is the control centre. And that's going to be sending signals to various parts of the body. For example, to the pancreas, which is responsible for producing insulin. Effectors muscles are going to do things like moving, for example, shivering. And then glands are going to be responsible for the production of other hormones. Control of blood glucose is very complicated. After a meal has been eaten, blood glucose levels start to rise. This is picked up by the pancreas. The pancreas produces insulin, which is sent out into the blood. The insulin in the bloodstream is going to cause body cells to start to remove glucose from the blood. Liver and muscle cells can take the glucose and convert it into glycogen and store it. Removing glucose from the blood will cause blood glucose levels to fall. If blood glucose levels get too low, this is also picked up by the pancreas. The pancreas will start to produce glucagon. The glucose that has previously been stored in muscle and liver cells starts to return to the blood. The most complicated part of this is getting all the names right. The stored form of glucose is glycogen. Glucagon will convert that into glucose. And this returning of the glucose will cause blood glucose levels to rise again. There are two different types of diabetes, type 1 and type 2. In type 1 diabetes, the pancreas doesn't work properly. So it doesn't produce the right amount of insulin. In type 2 diabetes, cells start to become insensitive to insulin. Symptoms for both are going to be loss of weight, an increased need to wee, being very thirsty, blurry vision, fatigue, so being very sleepy, and hunger. Treatment for type 1 diabetes is going to involve insulin injections. Type 2 diabetes, it's going to be controlling diet. Exercise. Well done, guys. Excellent work for making it this far. The rest is biology only. So if you're doing combined science, well done. You can go and have a relax or try some quickfire questions or go through the revision guide. DNA is a long strand of deoxyribonucleic acid, made up of lots of letters, A's, T's, C's and G's. And these twist round into a double helix. This double helix is still ridiculously long, so it further twists rounds so that it's in a chromosome. And this chromosome is located in the nucleus of a cell. Each 3-letter sequence of DNA is going to code for an amino acid. So here we have A, G, A. We start off with A, find G, then find A. So that DNA sequence is going to code into the amino acid arginine. The next 3 along, C, T, G, are going to code into leucine. And this will keep going until eventually we have a long amino acid chain. This can then fold up in very complicated ways until we get a protein that will look something like that. And proteins are responsible for basically everything that happens in your body. They are the hormones, they're the enzyme, they're the cell walls. Everything is a protein or dependent upon a protein. And these proteins are very, very specific. An enzyme substrate's active site is going to be very, very specific to the substrate. So if there is a mistake in our amino acid chain, if something is missing or if something is wrong, we put the wrong amino acid in there, then our enzyme, our protein, is going to fold up wrong. The mutation is going to have caused a change in the protein, which can then have a massive impact on how it functions. Meaning that it might not work properly, meaning that it might not break down what it's supposed to break down, meaning that it might not function in the correct way. There is a massive amount of DNA in each of our cells, and only some of it is useful. So say this section here might be non-coding, which basically means it's like junk DNA just getting in the way. Here we have our beautiful picture of the eye. The sclera, which is the white bit. The retina, which is where the image is focused. The optic nerve, which sends message to brain. The ciliary muscles, which change the shape of the lens. The cornea, which is a protective covering. Pupil lets light in. The lens is responsible for focus. And the suspensory ligaments hold the lens in place. If you are short-sighted, you can't see distant objects, and if you're long-sighted, you can't see close objects. In an eye that can see correctly, the lens will take the light and will focus the image on the retina. Whereas someone that is short-sighted, the image focuses before the retina, and someone that is long-sighted, the image focuses behind the retina. To correct short-sightedness, we need a diverging lens. And to correct long-sightedness, we need a converging lens. The brain is the control centre of the body. It makes sure everything functions properly and tells various different parts what to do. We have the cerebral cortex, the cerebellum, and the medulla. The brain is an incredibly complicated thing to study because for it to be functioning properly, it needs to be inside a living person. Doctors can work on mapping various different things by using MRI scanning and CT scanning, giving the person different stimuli to see which parts of the brain light up. The nervous system is incredibly complex and is overlaid on our spinal and muscular system. It consists of the brain, spinal cord, which together are going to make the central nervous system, or CNS, and all the neurons, the receptors and effectors. When you pick up stimuli, that signal needs to travel from wherever you picked it up, so your fingers, all the way up to your nervous system, your central nervous system. Sometimes just stopping at your spinal cord and then coming straight back again. That is going to be a reflex. This is going to happen when you touch something hot, you move your hand away without even thinking about it. Other times something is going to happen, the signal will go up to your brain, you'll think about it, and then you'll decide to move. The nerve cells involved in this are very long. So this cell body here is incredibly long. And this can send a fast electrical signal. However, when we come to transfer the signal from one nerve cell to another nerve cell, things slow down a bit because they have to cross a synapse. This is going to be a slow chemical signal. As the chemical has to be released, diffuse across the channel and then be picked up and then initiate another electrical signal. Phototropism means something is going to grow towards the light. Geotropism or gravitropism means something is going to grow towards gravity. Meaning your roots are always going to go downwards and your shoots are always going to go upwards. Gibberellins are important for growth. Ethene is important for ripening plants, and auxins are important for growth, and they're going to do growth in the right direction. Body temperature is going to be regulated by the thermoregulatory centre in the brain. If you are too cold, the hairs on your body will stand up. This is to trap a layer of air. You're going to stop sweating. Vasoconstriction will start. So your blood vessels will constrict so that they're further away from the skin. Less blood is going to flow close to the surface of the skin, so less heat is going to be lost from it. And your muscles are going to start to shiver, and movement is going to produce energy. If you are too hot, your hairs are going to lie flat, so they're not trapping any air. You're going to start sweating, and the water is going to evaporate, leading to heat and energy loss. And your blood vessels are going to undergo vasodilation, meaning that you're going to get wider so that blood can flow closer to the surface of the skin so that heat can be lost. The kidneys have 3 functions. They remove urea, they control the iron content, and they control the water content of the blood. There are 3 ways we can lose water from our body: in urine, in sweat, and when we breathe out. It's important to control the level of water in the body, because there's too much water. Is too much water taken up by cells by osmosis, then they might pop, or if there's not enough water, then the enzymes, the functions, the reactions won't be able to take place. There are 3 steps to the way that the kidneys function: ultrafiltration, reabsorption, and then the release. Blood enters the kidneys under high pressure, and water, ions, urea and sugar are going to be squeezed out into the capsule, which is at the start of the nephron. As this all flows along the nephron, useful things are reabsorbed. All of the sugar is going to be reabsorbed via active transport. Some ions, the amount of ions that we need, the type of ions that we need, are going to be reabsorbed by active transports. And enough water that we need is going to be reabsorbed. The hormone that controls how much water is going to be absorbed is ADH, which is antidiuretic hormone. And then anything that isn't reabsorbed is going to come out as wee. If kidneys aren't working properly, a person can undergo kidney dialysis. The dialysis machine will take over the function of the kidneys, but it is very time consuming. It takes about 4 hours and has to be done 3 times a week. So this has a huge impact on someone's life and is not a lot of fun. An alternative to dialysis could be a kidney transplant, but these come with very long waiting lists and there is always the risk of rejection.